Hello, everyone, and welcome to week 4, where we're going to be going over chapter 5. Um, Chapter 5 is going to be talking about the research methods of biopsychology. And there are a lot of methods that are used by biopsychologists. We will not be going through all of them, but by the time we get through this chapter, um, you're probably going to think that we actually did, because there are a lot of different ways for us to explore the science of biopsychology. Let's go ahead and get started. We're going to begin here with contrast x-rays. Many of you have probably had an x-ray done one time or the other, and you'll notice that they really don't focus on the function. They're just looking at the structure of something. And so that's not going to work very well for studying the brain. And so in this case, what we're using are contrast x-rays. Okay, so that there's a chemical that's injected probably into an artery and sent up into the brain area. And so then when the x-rays focus on this, they're absorbed at different rates depending on if that chemical is present or not. And that process is called the cerebral angiography. And here's a picture of a cerebral angiogram. Okay, so that's showing you that the chemical is going up through there. Basically, what you're looking at are the blood vessels surrounding the brain. So nothing on function, nothing really on structure. You've seen much more vivid pictures of the human brain using other methods. This is just kind of a rudimentary method for looking at the brain and looking at blood vessels, structural things. There's also the X-ray computed tomography. And again, it's a computer-assisted X-ray for visualizing the brain. And it provides a 3D representation of the brain. And I don't believe that I have one of those for you. Here's this just shows you what it does is that it's taking it uses x-rays to create a CT scan of the brain. So you can see those horizontal CT scans. It's taking one picture after the other, and then it kind of fits them together into a three-dimensional construction. So the computer is taking a two-dimensional photograph, and then over a period of time with many of these scans, it puts them together into a three-dimensional picture. Again, both of those are going to be the X-ray computed tomography is definitely a little bit more advanced than that cerebral angiography. But again, it depends on what you're doing. If you're looking at the blood vessels, the cerebral angiograph is going to be fine. Computed tomography is going to aid you in looking at the brain structures a little bit more, not in vivid detail, but better. And neither one of those are looking at function at all. Okay. Then we look at the magnetic resonance imaging. Now these are high-resolution images, and they're constructed from measurement of waves emitted by hydrogen atoms that have been activated within a magnetic field. And as you can tell, both of these are MRIs, and you can see how there's much more detail. You can look into that mid-sagittal slice there, and you can see the cortex, the cerebrum. You can see in the posterior area the cerebellum. You can see the brain stem, the corpus callosum. You can tell where tell the anterior of the brain versus the posterior, the dorsal ventral. It's a very vivid picture. Okay. And then we also have positron emission tomography, and these provide an image of brain activity. Okay, so what we've been looking at, the three that we've talked about so far, have all been just structural, nothing functional. But with a PET scan, The scan, the image of levels of radioactivity in various parts of one horizontal level of the brain. A radio-labeled substance is administered prior to the scan. That substance is the 2-DG is what they call it. It's 2-deoxyglucose. So it's very similar to glucose, which means it's going to be absorbed by neurons that are active. So if you think about that, you put the chemical in there, absorbed by active neurons, and then it's color-coded. So now we're able to see, and let me go to the next pictures that show where the activity, where most of the activity is occurring in the brain. So if you look on the left side, it says when the volunteer's eyes were either open on the left or closed on the right. Areas of high activity are indicated by reds and yellows. So remember, we're now going to be fitting all this stuff together. Everything that you learned in neuroanatomy, chapter 3, neurophysiology, chapter 4, we're going to build on it now. So now you're given a figure, and you're just expected to know lateral, medial, anterior, posterior, dorsal, ventral, right? All of those things that I mentioned to you at the time. And if you need to go back to that information, please do so. All of the e-lectures are posted, and there's no date stamp on there, so you can watch them as many times as you need to. But getting back to the figure 5.6 here, you can see that in the posterior area of the brain, where those arrows are pointing to the occipital lobe. Remember, the primary function of the occipital lobe was for vision. So on the left-hand side, the volunteer's eyes were open, so the occipital lobe was processing information. On the right side, they closed their eyes. And so now you see the reds and the whites disappeared. Okay. And then we talk about the functional MRI. So we already talked about an MRI, but now a functional MRI provides images of brain structure and activity. So it's going to help us with function as well. Okay. Uses a strong magnetic field. The structure is imaged using waves emitted by hydrogen ions. Same as what we learned. But the function is imaged using the signal created by interaction between oxygen and iron in the blood. Again, that is the same. The difference here is that there are many MRIs that are done in succession with one another very quickly. And so, again, kind of like that tomography where it's adding up individual pictures and it makes a three-dimensional. So here you have a functional MRI. The image illustrates the areas of cortex that become more active when the volunteers observed strings of letters and were asked to specify which strings were words. In the control conditions, subjects viewed strings of asterisks. This functional MRI illustrates surface activity, but images of sections through the brain can also be displayed. Okay. So you're looking here. Now, this individual, this brain here would be facing the right, because you can see the cerebellum at the bottom there. And so you'd be looking at the upper portion of the temporal lobe there. And you can kind of look at some of the central fissure and lateral fissure. You can see some of those characteristics. Okay. Going on to other techniques. We have the magnetoencephalography, the MEG. And this is a measure of neural activity, measures changes in magnetic fields on the surface of the scalp. Okay. So now you're looking at neural activity from scalp electrodes. And we're going to be talking about other techniques such as this also when we go into the EEG, which I believe is next. Yeah, the author doesn't go into MEGs very much. Produced by changes in underlying patterns of neural activity, and he leaves it at that. Okay. We also talk about the transcranial magnetic stimulation. It's not a measure of neural activity, but provides an experimental probe to alter neural activity. Okay, so through a strong magnetic field, we can either activate or deactivate brain structures, and therefore observe the changes in behavior. So through a strong magnetic field, the brain is actually altered in some way to activate or deactivate a brain structure, and then we can observe the behavior that is due to that activation. Okay. Now, EEGs are something that you might be a little bit more familiar with. Okay, electroencephalography (EEG) measures gross electrical activity of the brain, uses electrodes attached to the scalp. For the most part, these EEGs are going to be scalp electrodes. They use a little cream, they put it on the scalp. There's nothing invasive. Okay, they're not placed into the skull itself or anything like that. We are going to talk about an invasive EEG in a minute, and those actually are. Most of the time, also EEGs now are done with a it's kind of like a cotton cap, and the cap is put over with a little strap, and then all the electrodes are sewn into the cap. It just makes it a lot easier. I actually performed my master's thesis using a QEEG, a quantitative electroencephalography, which just meant that the scalp electrodes, everything was the same. It just uses a computer software to render the EEG recordings, as opposed to a series of pins like you would see in your book, like on figure 5.9. Okay. But there's many different techniques of EEG, many things that they're looking at, but you have to remember the one word here, gross electrical activity. So it's very, very hard for us to say that, okay, that electrical activity is occurring from the caudate, from the nucleus accumbens. Okay, we can't say that. These are scalp electrodes. All we're looking at is the combination of neural activity. Okay. What we can look at are brain waves and brain activity. So I think there's a couple of these in a minute. I'll talk about alpha waves and so on. But I don't want to go into a lot of detail actually in the alpha waves and so on, because we discussed that in vivid detail in our chapter on sleep and so on. The event-related potentials, now these are ERPs, and they measure activity accompanying a psychological event. And there's an example of that in a minute too. Okay, so here's some typical electroencephalograms and their psychological correlates. So an aroused state at the top, that's going to be a beta rhythm. That's what you see there. Relaxed is alpha, asleep is theta, and then deep sleep are going to be your delta. Okay, like I said, I'm not going to go into a lot of detail because we will in subsequent chapters. But these lines, they come from an EEG, and that's what's important for now. Now these are ERPs, and you can see that the evoked potential, the column on the right, that's when the potential was evoked, and you can see that there's a change in the EEG, and in this case it was a clicking noise. So the clicking noise was the evoked potential, caused a change in the EEG. Okay. Other things that we can study is muscle tension. We can do electromyography, or EMGs. This is a technique of measuring the electrical activity of muscles. So remember, we talked a little bit about how acetylcholine was responsible for muscle movement and that kind of a thing. So those, again, these are neurons that are activated at muscle fiber sites, and so these electrodes are picking that up. When I worked at the Chronic Pain Center, I used to work with EMGs a lot, we would look at, we would place them on the shoulders, sometimes the frontalis muscle on the forehead, sometimes around the neck area, just to see if people were bracing, what we called bracing. So they're in pain, they've been in pain for quite some time, and they don't even realize how they've altered kind of their stature and their posture because of this constant pain. And using EMGs, they were able to have that information fed back to them so that they could help themselves relax and so on. Okay, a raw EMG is going to be up here at the top of this graphic, figure 5.12, and then an integrated EMG is at the bottom. Integrated just means that through a computer process, it's taking all of those kind of mathematically highs and lows and creating one line to show you that obviously there was some kind of a muscle activation, maybe a clenching or something, and it caused in the top graph for the red to broaden, and then for the bottom one there, the line to heighten during that clenching. Okay, we can also study eye movement, which is electrooculography, or EOG. And this indicates the changes in electrical potential between the front and back of the eyeball. So again, if somebody is relaxed, you could help them with that. You could also, they oftentimes hook up an EOG during sleep studies. Something else that we'll be talking about in this class is rapid eye movement, which happens in a stage of sleep. And so we can definitely tell when that stage of sleep is initiated because of the eye movements, and we are detecting that. You can see the eye actually move under the eyelid, but this is a much better tool to use also for those small eye movements that we might not be able to see. Okay, what about skin conductance? Now, skin conductance is one of those feedback mechanisms because it's used also for biofeedback. But it's one of those that we don't understand real well. All we know is that when people are anxious or fearful, they seem to have clammy hands. Okay, and so that's what this is picking up. Measurement of skin conductance level is SCL, and skin conductance response, or SCR. Okay, so skin conductance, you would assume that somebody who is in pain, anxious, might be sweating more from the palms of their hands, and so this level would be higher. If you can get them to relax, then less sweat, less supposed anxiety. Cardiovascular activity, often used to look at physiological changes. It measures heart rate, blood pressure, and blood volume. And there's actually a tool that we use for this, which is the photoplethysmograph. The photoplethysmograph, and it just goes around the end of a finger, and it shines a light into the finger, and what it's looking at is blood flow. So again, in a relaxed individual, the blood flow is really good and plentiful in the fingers. In somebody who is bracing, anxious, nervous, scared, fearful, there's going to be less blood flow. Okay. Now we're definitely going to change gears here, and we go from non-invasive. Now remember, we did talk about a couple invasive. The PET scan, okay, the angiogram, those are things that require a chemical to be introduced into the system. So those are going to be invasive also. But we talked about many non-invasive. Your x-rays, your MRIs, your functional MRIs. We talked about the EMGs, EOGs, and skin conductance. Those are non-invasive techniques. Now what we're looking at here are very much invasive research methods. Primarily these are going to be done on animal species. Well, I should say they're always going to be done on animal species for research purposes, and then for medical purposes, for removing tumors and that kind of thing, of course, from humans, then we would also use these techniques. Okay. So let's start off with our stereotaxic surgery. Okay. It requires a stereotaxic atlas and an instrument. And these will be shown to you in just a minute in a picture. And I'll go back to that when we have the picture in front of us. Let's look at the lesions real quick. You have bilateral and unilateral lesions. Okay. Now let's go through our nomenclature again a little bit. Bilateral is going to mean that it's affecting both sides of the brain. Unilateral, just one. Most of the time when lesions are done for some kind of a disorder, they're mostly done bilaterally because the brain connects, that corpus callosum connects the two hemispheres, and so when that functionality is lost in one hemisphere, the other hemisphere picks up on that a lot of times. Okay, so this is also true for some of these disorders that you'll have to lesion, both sides if you want it to remediate. Okay. Several procedures requiring careful interpretation of effects. You have aspiration. Aspiration just means that it's being vacuumed. So they would put a cannula into the brain and actually vacuum out the brain tissue. Radio frequency, is really high frequency, so it actually burns the neural tissue. Knife, of course, that's just cutting it, just like you would in doing something orthoscopically in the knee, where you're going in there and you're cutting out the cartilage. Just going to be much, much finer tools and using a lot more microscopy. And then there's reversible lesions also. And these are the ones for in case of like cryogenically. You can actually freeze an area. And what that means is that you freeze an area, you identify the behavioral effects of that, loss of language, loss of interpretation of language, whatever it might be, and then once the tissue is warmed back up, it's reversible. So it's not a permanent lesion, which is really amazing for some of the surgeries and stuff that they do. And I think I'll get back to that in a minute. Okay, here is your stereotaxic surgery. Up at the top, you have the atlas. This is just going to be a book. You just lay the book in front of you, and you have brain slices. Okay, and so you flip through the book until you find the brain structure that you're looking for. There's a term called bregma. Bregma is at the very top centermost point of the brain. So you can start at bregma, and then the atlas will tell you, okay, you need to move, like in this case, 2.8 millimeters posterior, 4.5 millimeters lateral. And guess what? You're doing stereotaxic surgery, and this could easily be a human also. They would use the same process. And imagine if you got your posterior, anterior, lateral, medial mixed up. It would be very, very detrimental to the animal or your patient. So again, it's important to understand the directionality of these. Okay. Here is an example of a lesion, and the lesion is being performed through a knife, and that little blade pops out from the cannula there and then can cut a very small area of the brain. Okay. And I think your author goes into this a little bit when he talks about we would like to think that we put this cannula down in here and use aspiration, radiofrequency, knife, whatever it might be, and we cut out this little section. Okay. Is that all the damage that we did to this brain? And the answer there would be no, because we have this instrument being fed through all of this neural tissue here. Okay. So in some cases there is damage due to that as well. We try to minimize it as much as possible, but it does happen. Okay. All right. Here's a little bit more. Lesioning can be used to remove damage or inactivate a structure. The electrical stimulation can be used to activate it, and stimulation of a structure may have an effect opposite to that seen when the structure is lesioned. Okay. Those are just some characteristics to our lesioning technique. Okay. All right. Some more invasive research methods. What about those where we're using the recordings? Okay. Intracellular unit recording. This would be placing a cannula into the brain, into the cerebral cortex, or maybe even deeper, and actually penetrating one neuron. Okay, membrane potential of a neuron. Extracellular would be recording the firing of a neuron. I guess yeah, you would put the cannula down into the inside of the neuron would be intracellular. On the outside of the neuron would be extracellular. Okay, so both of those would be for recording a neuron. Multiple unit recordings would be firing of many neurons. So you would be interested in many neurons. The multi-unit recording, it wouldn't require nearly as small of a cannula, right? Because you're not trying to penetrate just one neuron, but put it into the extracellular fluid around many neurons. And then there's the invasive EEG. I mentioned to you that I was going to come back to that. And these are actually electrodes that are placed into the skull with little screws. Okay, but you can imagine that the purpose for that is to get a better idea of the neural activity. Remember in chapter 3, neuroanatomy, we talked about the meninges, we talked about cerebral spinal fluid, we talked about the skull. There are several protective layers around the brain. So the closer we can get to the brain, the more detailed information we can receive about the neural activity. Here are some examples for you. In intracellular unit recording, top left, and then at the bottom, an extracellular unit recording. Multiple unit recording, that's surrounding around, see it says a number of action potentials. And then invasive EEG, again, you're going to have that line. This looks like it's kind of a combination between beta and alpha rhythms, but it's going to be more specific to a activity. So if you have the individual close their eyes or speak or something like that, and one of those areas light up, like occipital lobe, visual cortex, you're going to get a better picture using it this way than just through those scalp electrodes. And just to give you a little example again, or a little clarity for those reversible lesions. Here's something where, let's say an individual comes in, they have a tumor. The tumor is very close to their language center. Most individuals, most humans, their language center is in the left hemisphere. Let's say that the tumor is in the left hemisphere, okay, around that language center. So what they would do is they would actually cryogenically, or they could use it through chemicals also, lesion that area. Okay, if the individual loses their ability to speak or interpret language, then we know that if we remove this tumor, we could be removing their ability to interpret language or speak. Okay, if we do the cryogenic lesion in the left hemisphere and nothing happens to their speech, then we can, oh, well, you know what? Even though most of the time the speech area is in the left hemisphere, for some individuals it is in the right hemisphere. Theirs happens to be in the right hemisphere because they can still speak or interpret language. So we're good to go. We can go in there, we can take this tumor out without any damage. Okay. So that is a huge benefit for reversible lesions. Okay, measuring chemical activity. We already talked about 2-deoxyglucose because that was in relation to PET scans. Okay. Use autoradiography to see where radioactivity accumulates in brain slices. The difference here is that this is going to be entered into a subject, and then we're going to take that brain out. We're going to slice the brain and then find out where the this glucose substance was absorbed. Cerebral dialysis measures extracellular concentration of specific chemicals in live animals. Let's look at a picture here. So here, again, 2-deoxyglucose technique. Subject was injected with radioactive 2DG. Then for 45 minutes, it viewed brightly illuminated black and white stripes through its left eye while its right eye was covered. Because the ground squirrel visual system is largely crossed, most of the radioactivity accumulated in the visual structure of the right hemisphere. Okay, we'll talk more about contralateral matter and that kind of thing in subsequent chapters. But again, you're looking at visual cortex being associated. Occipital lobe, visual cortex is in the occipital lobe, and it's all about vision. There are also ways of locating neurotransmitters and receptors in the brain. Remember, we talked about those stains, Nissl stains, Golgi stains, that kind of thing. This is similar. Dye or radioactive labels used to visualize the protein of interest. The term here is immunocytochemistry. It's based on the binding of labeled protein-specific antibodies. So something foreign is introduced into the system, and immediately the system starts creating antibodies because it sees this as a foreign substance. Okay? These antibodies are created and then bind and remove and destroy antigens or foreign proteins. So when the antibodies attach, we can track those. So here's an example of immunocytochemistry. A rat's substantia nigra reveals dopaminergic neurons. Okay, remember dopaminergic, so those are going to be receptors for dopamine, dopaminergic neurons that have taken up the antibody for tyrosine hydroxylase, which is an enzyme. Anything that ends in ASE is an enzyme. Okay. So this is a very good descriptive picture here, showing those neurons. Okay. And then here's one, the in situ hybridization. This color-coded frontal section through a rat brain reveals high concentrations of mRNA expression for an endorphin in the striatum. And I didn't go into a lot of detail on this one specifically with the mRNA and so on, but just to show you the difference between that and the immunocytochemistry. Okay. Now, another thing that's being studied quite significantly in many of these physiological psychology labs around the nation is this gene knockout technique. Okay. And the idea here is that subjects missing a given gene can provide insight into what the gene controls. Now, we have to be very careful with this because we know that this outward behavior is not due to just one gene. It's a combination of many, many genes. But we're able to now breed rats that do not have certain receptor sites. So let's say dopaminergic receptors. There's D1, D2, D3, D4. There's even more than that. But let's say that through genetics, we're able to eliminate D1 and D2. Okay? And then what we can do is identify or build pharmaceuticals, psychopharmaceuticals, that will only affect D3 and D4 and then leave D1, D2 alone. Anytime that we're dealing with psychopharmaceuticals, it is most important to build drugs that are very, very selective. We want the action of that drug, whether it is to destroy an enzyme or whatever it might be, but we wanted to do it in a very selective manner. Doing it throughout the entire brain causes a lot of these contraindications, these negative effects that you hear about. So the more specific the drug, the better. And there's also gene replacement techniques where we can insert a pathological human gene into mice and then try to cure that mouse from their ailments, which was produced from the pathological gene. Then we have the green fluorescent protein. Okay. These GFP genes can be inserted into the DNA of neurons, and then a color can be viewed when the targeted neuronal genes are expressed. And you have something called the brain bow here, and I have a that pretty impressive. Each neuron is labeled with a different color, and so you get this view of all of these neurons here. They don't tell us where this is at, but you got a lot of, it looks like a lot of motor neurons there because you have the cell bodies. It looks like they're multipolar neurons. Okay. And now we definitely switch gears once again. So we talked about non-invasive techniques, invasive techniques. We talked about doing studying the brain through electrochemical manner. We talked about it in just lesioning, even to the rudimentary idea of slicing or cutting or vacuuming out a piece of neural tissue. So we've talked about all those techniques. Now we're going to switch gears and talk more about clinical neuropsychology. Okay. So neuropsychological testing is very time consuming. It assists in diagnosing neural disorders, serves as a basis for counseling and caring, and provides information on effectiveness and side effects of treatment. You have to remember that, let's say that somebody has a intracranial bleed or they have some kind of a brain damage. You're going to have, depending on the severity of the ailment, you have the neurosurgeon. The neurosurgeon goes in, fixes the bleed, fixes whatever they have to do. Then they're probably going to be seen by a neurologist who will be looking at the brainwave activities and sleep patterns and that kind of thing. And then they'll be done with that. Okay. But then comes the clinical neuropsych. And the clinical neuropsych can really delve into much deeper into ailments that the individual still might have due to this injury, and some of those being maybe memory or depth perception or language or something like that. So that is what we're looking at. Oftentimes the clinical neuropsychologist will use a single test approach, and this is used to differentiate brain damage from functional or psychological causes. The single test approach is normally used with a standardized battery of tests, and the most common one for that would be the Halsted-Reitan. And many clinical neuropsychologists will also customize this battery of tests to the specific needs of their client. Okay, and so then it would be a customized test battery. So what would they use? Well, they almost always use some kind of an intelligence test, the WAIS. They probably will use digit span for memory. They will look at language issues, and they will also look at language lateralization. And here again, the sodium amytal used to anesthetize one hemisphere. That's what I was referring to you. Also, that would be a chemical reversible lesion. Just like that, cryogenic would be freezing. This would be a chemical reversible lesion. Or the dichotic listening, which what they do is that the individual puts on a pair of headphones, and then in one ear they're provided 5 numbers. Let's say 5 numbers to the left ear, and then 5 numbers, but different numbers to the right ear. And then they ask them, how many numbers did you hear? The individual will respond to more of the numbers in the ear, which is more primary for them. Okay. So the dominant hemisphere shows superior hearing ability. Okay. And so again, that's just able to find out, okay, language lateralization. Are the language centers more in the left hemisphere or in the right hemisphere? Okay. These neuropsychologists are also going to be looking at memory. I didn't want to go into a lot of detail on this because we have a whole entire chapter on memory also. But the difference between short and long term. Anterior grade meaning before the after the accident, retrograde meaning before the accident, semantic meaning the meaning of memory, episodic, the episode, event and meaning, explicit what you can relate to, implicit what you is kind of inferred. Okay. Language again, phonology, syntax is also listed there. And then there's frontal lobe functioning. So this is going to be more of a higher order cognitive task. Okay, the Wisconsin card sorting task, you can see it in the book. Oh, actually, there's a diagram here. And what it asks of the patient is to sort the cards based upon color, and then after they start making mistakes, they change it to sort based upon shape and so on. And if they have a hard time sorting those cards, maybe they sort by shape and then they can't, even though they're getting errors and errors and errors, they're not switching it to, oh yeah, that's right, maybe I should be sorting them based upon the color. And so those can definitely show us brain ailments or deficiencies. Okay, also in cognitive neuroscience, the belief here is that each complex cognitive process results from the combined activity of simple cognitive processes. Each complex cognitive process is mediated by neural activity in a particular area of the brain. So again, through all of our more advanced brain scans and so on, we're able to see the inner workings of the brain and how it is responding to learning or to the environment in some way. Okay, the goal is to identify the parts of the brain that mediate various constituent cognitive processes, and oftentimes can be paired with PET scans and functional MRI images, okay, during the cognitive tasks. Now here's a concept that your author comes up with also. It's commonly employed in cognitive neuroscience. What they do is, if you look at the diagram there real closely, on the top left it says visual stimulation, then the image just to the right says control, and then the one furthest right says difference image. So what we're looking at here is this could just be a PET scan. And so they're looking at the difference between visual stimulation and control, and that difference is what you see in the image on the furthest to the right. So it says if the activity that occurred when the subject stared at a blank screen is subtracted, it becomes apparent that the perception of the flashing checkerboard pattern was associated with an increase in activity that was largely restricted to the occipital lobe. Again, occipital lobe, primary visual cortex, so it's all going to be about vision. Okay, some paradigms for studying animal behavior. The open field test. What this is referring to is that you try to put the subject into an environment that isn't as restrictive as a cage or something like that. It's probably not going to be the whole world out there, but it might be a very small, maybe 10 by 12 piece of dirt with some vegetation around there and everything. It makes the appearance of more of the natural environment. That's an open field test. They can look at tests of aggression and defensive behavior, tests of sexual behavior, and we'll see some diagrams of that in just a second here. Also, oftentimes animal behavior is studied through learning techniques or conditioning techniques. Everybody knows about Pavlov and the salivating dogs, etc. But operant conditioning is also very important, because what we can do in the last term there, that self-stimulation, is that we can actually train or condition a rat to select a bar to receive some kind of a rewarding effect. Okay, so what we'll do is we'll put a rat into a Skinner box, and we'll condition that rat to, when you press that lever, you will be electrically stimulated in the area of the brain where the reward pathways are. Okay. That reward pathway is the same as it is in humans for our reason for hunger, our reason or seeking out food, seeking out water, etc. Okay, and we have a whole nother chapter on that when we talk about drugs and addiction. Okay, but it's important to know that we have to condition the animal to respond to this operant conditioning, pressing that bar to study the animal behavior, and in this case, study addiction. Semi-natural animal learning paradigms, I talked about more of an open field idea, but taste aversion is a good one too. This is where an animal is given an emetic, right, which is a drug that causes illness, and then the predator will stay away from that animal. They do this on a regular basis at some of the national parks, I should say, around the nation, where they're trying to avoid, coyotes or wolves from eating livestock and that kind of thing. So once they eat that animal and they get sick from it, they normally stay away. Also, you have the radial arm maze, and this is specifically used for spatial learning. I think I have a picture of these coming up here pretty soon. The Morris water maze is, you can think of that as like a barrel. It's kind of milky water there. Put the rat inside. He swims around, and just under the surface of the water is a little stool. Once he finds that, he props himself up out of the water. So what you're doing is you're taking away kind of visual cues, and you're looking at spatial learning and how they navigate in their environment. And we can actually look at removal, again, lesioning certain areas of the brain, and see if how this is affected, how their spatial learning is affected. Conditioned defensive burying. Following a single aversive stimulus delivered from an object, rats will spray bedding at the object, and then anti-anxiety drugs decrease the amount of burying behavior. So here's another one. Another example is whether it's the operant conditioning, that pressing the bar, or if it's this defensive burying. If you provide drugs to the subject, which are like anxiolytics, anti-anxiety drugs, would that prevent the burying? Would that relax the subject so that they're not pressing the bar as many times? So that's what they're looking at. And here is just an example of a radial arm maze viewing spatial learning. As you can see, only some of these radial arms actually have reward in there. You put the rat in there. After a couple trials, he will go just to those radial arms that had the food source, and he'll avoid those empty. And then here's an example of a rat. Obviously, it's a fear response, and so they have a tendency of burying themselves. Okay. Again, provide a drug into this individual, provide that same stimulus, which would have that cause that fearful state, but then with the drug, if they didn't care about that stimulus nearly as much, you wouldn't see that behavior brought out.